Now let's continue on with our discussion of biotechnology. We've just finished discussing PCR and the purpose of it, which is to amplify or make large amounts of copies of DNA. Now let's talk about how it is exactly, once we have a large amount of DNA copied, how do we take that DNA then and use it to manufacture what's called a DNA fingerprint? Let's take a look at this process, which utilizes something called an electrophoresis machine, and actually you're going to get to do this in lab in a week or two yourselves. This gel electrophoresis process I'm going to go through in just a second, and involves a contraption that you can see in this picture below. It's about the size of two of your hands put together. It's not a very big device. What happens in gel electrophoresis is I take all that DNA I just copied. So I've got my million or so copies of the DNA I'm after. And the first thing that I do is I expose that DNA to something called restriction enzymes. Restriction enzymes are enzymes that bacteria naturally produce, and the bacteria naturally produce these restriction enzymes to combat viral infection. But we're taking those restriction enzymes and using them for a different purpose. Restriction enzymes are unusual in that they have the ability to chop up DNA into smaller pieces, and they do so at very specific locations. They do this by looking for a particular region that they're sensitive to, and each different type of restriction enzyme is sensitive to a different DNA sequence that it will cut at. If we look at the restriction enzyme called EcoRI, which is one of the restriction enzymes that we found in the E. coli bacterium, it is very sensitive to the sequence GAATTC. And anytime it sees the sequence GAATTC in a DNA molecule, it will cut at that specific location for that particular sequence. And the way that it cuts is very unusual. Notice that it doesn't just make a straight up and down cut like this, but rather it zigzags inward this way. So that what I end up with looks like this below, where I have two dangling ends of DNA that we call sticky ends. We call them sticky ends because they will readily anneal with any other DNA that has the right sequence to stick with it. Now again, different restriction enzymes will be sensitive to different sequences found in the DNA molecule, and I've only shown you just one of them in this example. Now, why do I cut up the DNA? Well, because by cutting up the DNA, I get different lengths of DNA present as a result, and different individuals, since all of our DNA varies just slightly, those restriction enzymes may cut in slightly different locations on different people's DNA. And as a result, the DNA that we get chopped up with the restriction enzyme will have different lengths to it depending on the person. Now we take that DNA, all of those different lengths of DNA that we've just chopped up, and we suck them up into a little pipette here, and then we do what's called loading the gel. And this simply means we take agarose gel. Agarose gel is that jelly-like substance that we use in Petri plates for cultivating or culturing bacteria. And we take and inject that DNA into a tiny well or a tiny little dip at one end of the agarose gel. And you can see this individual down here doing this. The dark little bands in this area represent the different regions of DNA that have been dropped into individual wells in that gel agarose. Again, you'll get to do this in lab, so you'll get to see firsthand how this is done. Now, we take all the DNA. Let's say we have 3 suspects, and we have the DNA from the scene of the crime. We would take the DNA from the scene of the crime, load it into one well, and then we would take the DNA from suspect 1 and load it into another well, and the DNA from suspect 2 and load it into another well, and the DNA from suspect 3 and load it into another well. Then what happens is we are going to run an electrical current through the gel to move the DNA. We put the DNA near what's called the anode, which you'll remember from chemistry is the end of the electrical current that, or the node of the electric current that has a negative charge to it. DNA overall itself has a negative charge, just like proteins do. So if we load the DNA at the negative end of the gel, and it has a negative charge, as we run a current through it, the DNA is going to want to move towards the positively charged cathode at the other end of the gel. Okay, so the negatively charged DNA is going to be attracted to the positive end of the gel where the cathode is located, and the DNA moves in that direction. What's interesting about this is, remember, I chopped up the DNA into all these pieces of varying lengths using those restriction enzymes. Those different lengths of DNA will travel at different rates. The bigger pieces of DNA have a harder time moving through the gel agarose, and as a result, they will not travel as far. By comparison, the smaller fragments of DNA will be lighter and smaller and will more easily move through the gel towards the positive cathode. So once we turn on the electricity and let it run, the small pieces tend to run across the gel towards the positive end more rapidly than those that are larger. And by the time we get all of the DNA spread out and we turn off the current, if we then stain the DNA so that we can actually see it, we would see that, again, as I said, small segments travel far, large segments travel a shorter distance. And we get a separation of the segments in the production of these banding patterns. Again, since you guys get to do this in lab, you'll have a first-hand experience with this. And once we have these bands, then we can do comparisons, as I did previously, to show how a particular parent might be the father of the children, or a particular suspect might be the one that actually perpetrated a particular crime. Okay, so that's kind of the end of our discussion on gel electrophoresis. I'm going to move on now and talk about a different topic called gene therapy. Gene therapy, remember again, is the use of genetics and the manipulation of human genetics in order to treat diseases. One of the ways that we can use gene therapy is to replace defective genes in a patient with functional versions of the same gene. So I'll give you an example. Cystic fibrosis is a human disease that involves cells of the lungs and the pancreas not being able to properly manufacture mucus. The mucus that they manufacture is extremely sticky and thick, and it's not watery at all. And as a result, it tends to stay lodged in the lungs and does not move up out of the lungs via coughing very readily. It used to be that individuals with cystic fibrosis, and this is an inherited disease due to a defective gene that does not program for the proper manufacture of mucus, it used to be these individuals were lucky if they made it to age 21. Lucky. But different types of medical treatments today are allowing the lives of cystic fibrosis patients to extend quite a bit. Now, cystic fibrosis is an autosomal recessive disease. Remember, autosomal, again, means that this gene is found on a non-sex chromosome, and recessive means that in order to have the disease, I would have to have 2 copies of the recessive allele present. How do we use gene therapy to treat cystic fibrosis? Well, in this case, because the defective gene in question is one that programs for the proper manufacture of mucus, what doctors have been able to successfully do in the past few years is to take a virus that is harmful to humans but does infect humans, and we inserted a proper copy of the mucus gene, one that codes for the proper manufacture of mucus. We inserted that into our virus. So here's our virus, and into the viral DNA, we have, or in this case, the viral RNA, we have inserted messenger RNA that codes for the proper manufacture of mucus. Then we put that into an inhalant, and we allow the cystic fibrosis patient to inhale this virus into their lungs. The virus infects the cells of the lungs, and again, remember, this is a harmless virus to the patient. It doesn't cause any symptoms or disease. But what it does do is it inserts that proper copy for the manufacture of the mucus gene into the cell of the lung of the patient that has cystic fibrosis. So what we in effect do is introduce a proper copy of the mucus gene into the cystic fibrosis patient so that the patient can start making proper mucus in their lungs. Now, they've already done this a few times. Medical researchers have done this successfully with some patients. Their success rate isn't super tremendous. They had about a 40% success rate with the patients they tried this inhalant on, but it's encouraging that this has worked this much, and with further tweaking and manipulation, they could probably increase the success rate. They do notice that after several months, the patient has to take another inhalant again to reintroduce the gene into their lungs, but it is an effective way to treat a human disease using genes inserted into the human cell. Now I'm going to shift gears again, and I want to talk for a minute about something called GMOs, which stands for genetically modified organisms. Genetically modified organisms are plants or animals that have had their chromosomes or genes altered in some fashion to the benefit of man. It might be, as I gave an example in the previous lecture, a bacterial cell that's had the human gene for making insulin inserted into it, so the bacterium now makes insulin for us. That's a genetically modified organism. It could be a goat that's had a human gene inserted into it, so it now makes human growth hormone, or hemophilia proteins that combat hemophilia. It could be corn that's been engineered to be resistant to a particular virus or a particular insect or a particular herbicide. All of these are examples of genetically modified organisms. A great example that's one that's very worthwhile in recent years was the development of something called golden rice. Golden rice is basically a version of rice that has the ability to manufacture large quantities of vitamin A. And because it's been genetically engineered to manufacture large quantities of vitamin A, feeding golden rice as opposed to regular rice to individuals in poverty-stricken countries such as China and India helps to reduce the rate of, for example, blindness in those areas. People that are malnutrition and don't get enough vitamin A in their diet can go blind, for example, and this would help to prevent blindness. And it's something as simple as genetically modifying the rice. The rice farmer plants this rice seed instead of the normal rice seed. The rice grows the same. It has a slight yellowish color to it, which is why we call it golden rice. But the benefit is by genetically modifying that organism, we now have the ability to get vitamin A into the diet of these impoverished people in a manner that would never have been possible before. So how do we go about inserting a particular gene for, say, the manufacture of vitamin A into a plant, for example? Well, we can use lots of different ways to do this. One is to use something called a gene gun. I love this. This is a way of literally shooting the gene inside of a cell by putting the gene on little particles of tungsten, which is a type of metal, and then shooting them inside the cell, and some of those particles of DNA will get introduced into the nucleus of the cell. That's one option. Another one is to let a bacterial cell do the transferring for us. We could use bacteria that naturally infect rice, for example, and use them, let them introduce the gene for us. Or we could use a virus to do the same thing, a virus that naturally infects the rice. We could insert the gene into the virus and then let the virus insert it into the rice plant. Now, the last topic I want to talk about is one that I know you all have heard of, though you may not be super familiar with it, and that is the topic of stem cells. Stem cell research has been in the news and the political arena for years, and it's been a topic of great controversy because for a long time the only means by which we knew to be able to get a hold of stem cells for research in the laboratory was to harvest them from a human embryo. One that was usually in something like the 32 to 64 to 128 cell stage. So it was very, very early on, within a couple days after fertilization, that this was harvested. But once the stem cells were harvested, that spelled the end of the embryo. Now, what exactly are stem cells? Let's back up first and talk about that. Well, stem cells are special cells found in your body that have an ability to do two very unusual and unique things. Firstly, stem cells have the ability to divide endlessly. Normally, most cells in your body, most what we call somatic cells, or just regular old body cells in your body, are only allowed to divide about 50 times or so. And after about 50 cellular divisions, they are told to undergo a process called apoptosis, which means programmed cell death. It's kind of like cell suicide. And the reason for that is every time that your cell divides into two new cells, naturally there will be a few mutations that occur because DNA polymerase does not perfectly copy the DNA every single time. A couple mistakes will happen. By the time we get up to 50 divisions, there are enough mutations or errors present that it may start to affect some of the functioning of some of the genes on those chromosomes. So at that point, the cell is told to undergo apoptosis and die so that it doesn't pass on those mutations. Stem cells are different. Stem cells are allowed to divide endlessly without being told to stop. So that's the first major difference between stem cells and regular somatic cells. The second difference is this. If you remember with a regular somatic cell, when it undergoes mitosis, it gives rise to two identical daughter cells. The daughter cells are identical to each other, and they are identical to the parent cell they came from. Stem cells are different. Stem cells have the ability to give rise to two different types of cells. When a stem cell undergoes mitosis, it gives rise to another stem cell, as you would expect, but it also gives rise to something called a differentiated cell. This differentiated cell is a cell that has the ability to develop into something other than a stem cell. For example, I could have a stem cell that gives rise to different types of white blood cells in your body. That stem cell, when it divides, will produce another copy of itself, another stem cell, and then it will also produce a differentiated cell that has been told, you are no longer a stem cell. You must now develop and mature into a white blood cell. Okay? Now, there are different types of stem cells that I'm going to talk about on the next slide. The ultimate stem cell is a fertilized egg, or what we call a zygote. A zygote results from the union of an egg and sperm, and that stem cell, that single fertilized egg or zygote, is the ultimate stem cell because it has the ability, think about this, to give rise to every single cell in your body. That one cell produced the 50 trillion cells that are now called you. Not only did that one cell, that zygote, give rise to all the cells that are found in you, it would also have the ability to give rise to the placenta and the umbilical cord that attached you to your mother so you could receive nutrients while you were developing in the womb. Now, let me back up here for 1 second. I want to talk very quickly about the three types of stem cells. There's what is called the totipotent stem cell. Potent means power. Toti means all. So this is an all-powerful cell. And the only totipotent stem cell out there is the zygote, that fertilized egg. A totipotent stem cell has the ability to give rise to all of the cells in you, as well as placenta and umbilical cord, the whole 9 yards. Okay? After a few days of dividing, that zygote will be a few hundred cells large. And at that point, many of the cells will have become what is called pluripotent stem cells. Pluripotent stem cells have the ability to give rise to all cells in your body, but they cannot give rise to the placenta, and they cannot give rise to the umbilical cord. Other cells have already been told to do that job. So pluripotent stem cells can give rise to all the cells in your body. And it turns out that pluripotent stem cells are the ones that researchers harvest and try to get a hold of for research purposes. Stem cells are really attractive for research because they can give rise to, pluripotent stem cells are really attractive for research, I should say, because they have the ability to divide endlessly so we can sustain them in the lab for years and years and years, and because they have the ability to give rise to any of the other cells in your body, we can use them to study all kinds of different functions in the body without actually having to go to a human to do the studying. Now, the third type of stem cell is called totipotent stem cell. And at about week 6 to 8 of embryonic development is when we see the appearance of totipotent stem cells in an embryo. At this point, totipotent stem cells no longer have the ability to give rise to every cell in the body, but rather they only are able to give rise to cells within a specific lineage. So, for example, I could have a bone multipotent stem cell. It could give rise to all the different types of cells that are found in bone, but it wouldn't be able to give rise to a cell in your stomach, for example. I could have a blood totipotent stem cell. It would be able to give rise to red blood cells and white blood cells, but it wouldn't, for example, suddenly be able to give rise to a neuron, a nerve cell. Okay? Most cells in your body, remember again, are not stem cells. They are somatic or regular old body cells, which are also called unipotent cells because they only have one power, and that is to divide up to 50 times and then die. So again, our 3 types of stem cells are totipotent, which gives rise to all parts of you plus placenta and umbilical cord. Pluripotent stem cells that give rise to all parts of you but not to umbilical cord and placenta. And again, pluripotent are the ones we're after for research. And totipotent stem cells, which have the ability to give rise to all cells within a particular cell lineage, but not able to give rise to any cell in the body. Now, again, on this topic of why are stem cells so controversial, why are pluripotent stem cells so controversial? I said before they're useful for research because they divide endlessly, so we can sustain cell lines for long periods of time, years and years and years, in fact, and because they have the ability, the pluripotent ones have the ability to give rise to any cell in your body, we can make colonies of nerve cells, colonies of stomach cells, colonies of skin cells, colonies of cells found in your eye, for example, and use these for research purposes. So pluripotent stem cells are extremely attractive and sought after. The way that they, again, are harvested here is our developing embryo. At this point, it's called a blastocyst. This is within a couple days after fertilization, and there's an inner cell mass here that consists of those pluripotent stem cells. And a needle is used to extract that inner cell mass from the embryo and put that into a petri plate for reproduction of the cells, harvesting, etc., for lab use. Once we remove that inner mass of cells, the embryo dies. But that inner mass cell, remember again, because it is pluripotent, it can give rise to any kind of cell we want it to in the body, with the exception of the umbilical cord and placenta. So we can use those cells in the research of the skin, the nerves, the eyes, bones, blood, lungs, the lining of the gut, the liver, muscles, and every other kind of cell you can think of in the body. They're exceptionally useful for research. Now, there are some newer techniques that are doing a couple things. One is figuring out if there's ways that we can take multipotent stem cells, remember those are ones that can give rise to cells only within a lineage, and converting them back to pluripotent stem cells. The reason this would be attractive is we as adults have multipotent stem cells in our body, and wouldn't it be nice if we could just harvest some of our own multipotent stem cells from, say, our blood, and convert them into pluripotent stem cells, and then not have to go to this whole thing of harvesting pluripotent stem cells from an embryo. So this idea of harvesting from adults is an interesting one by converting the multipotent back to pluripotent stem cells. There's also some research that suggests there are some pluripotent stem cells still present in an adult, which we may be able to harvest and use directly. So that's the end of our discussion on biotechnology.